welcome everyone. I'm so glad that you can join us for this evening's program, Talk with the Scientist Marine Biologist, Dr. Lydia Baker. My name is Vivian, and I'm with the Tampa-Hillsborough County Public Library. This program is being presented as part of our summer reading program. We would like to thank our community partners for helping us bring you these great summer reading programs online and in branches. I am joined today by our guest speaker, marine biologist, Dr. Lydia Baker. Dr. Baker got her PhD from the University of Hawaii at Manawa and has researched animal-associated bacteria, specifically in marine ecosystems. She is currently a lecturer at the University of Miami in the Marine Biology and Ecology Department and is the head of the Masters of Professional Sciences Tropical Marine Ecosystems Management track. Now I'm going to turn things over to Dr. Baker.
Thank you so much, and thank you both and all for joining us and for the invitation to talk to you a little bit about my research and what's going on in that area. It's actually a pretty big, very wide variety of things that I work on, but they all kind of fall under the classification of bacteria and ocean life. All right, so yes, I work mostly with bacteria and ocean life, and that is widely encompassing. What I study are symbionts, and I think when people think about symbionts, they kind of think about, of course, Finding Nemo, right? There's the symbiosis that is between the clownfish and the anemone, where both are protecting each other, both mutually benefit. There's, of course, other examples in the macro world, including just even seeing bees interacting with flower, because along with eating pollen, they're spreading around the reproductive potential of flowers. So kind of thinking about everything encapsulated in this kind of blanket term of symbiosis can be helpful. However, there's lots of different kinds of symbiosis. So the thing that we think about most often when we think about symbiosis is a mutualism. That's where both organisms are benefiting each other. However, commensalism, just where one's benefiting and the other one just doesn't care, such as with cleaner fish and sharks, is an example of a commensalism. And then also, even though we don't like to think about it as being a symbiosis, a parasitism is also a kind of symbiosis. What we're just able to understand more about, just because we've advanced so far with molecular tools, is that we have so many examples of bacteria interacting with larger organisms, with eukaryotic organisms. And so what you're seeing on the upper left-hand side of your screen is actually bacteria that are interacting with tree nodules. And this is actually how we have plants that are able to produce proteins, because these bacteria are helping to enrich their environment by providing more nitrogen. But we also have cases like on the upper right-hand side, the Hawaiian bobtail squid. It uses bacteria to do counter shading, that is, to kind of cloak itself in the light of its bacteria, so it kind of looks more like moonlight. We also have bacteria that interact with flower nectar, and actually some bacteria can make flower nectar more appealing to pollinators. And of course, we have the bacteria that live in our guts that help to do lots of digestive processes. So the bacteria that I really focus on are actually part of all of these different organisms that you see here. On the upper left-hand side of your screen, those are diatoms. These are little tiny microscopic algae that are doing photosynthesis in anywhere that there is water. So your tap to the open ocean, bacteria are interacting with them and helping them to survive in these environments. I've also looked at, again, those bioluminescent bacteria, and I'll talk about that some today, both in the Hawaiian bobtail squid, but mostly in anglerfish, and to a lesser extent, flashlight fish. Flashlight fish are those little guys you see there with the little patch underneath their eye that's filled with luminescent bacteria, but also bacteria that interact with our coral here in Florida. So a lot of times when people think of bioluminescent symbiosis, they think of this gigantic anglerfish going after Nemo and Dory. All right, that's not Nemo, that's the clownfish, I forget his name. Anyways, they're actually quite cute, I think. They're these big, huge goo balls with these googly looking eyes. This is a real anglerfish. This is actually the second most common anglerfish in the ocean. And that little tiny thing on the top of her head is a lure, and it contains bioluminescent bacteria. Now, most people don't know this, but anglerfish are actually the most speciose, that is, the most different types of species exist in the deep sea. And so here's just a subset of the different genus of them. So different, we always qualify down. So these are 6 different species as well. And you can see right away that they are very different sizes and morphologies, but they all have these little lures on the top of their head. So because we know they're lit with bacteria, and because anglerfish diversified about the same time that dinosaurs roamed the earth, so about that time, one type of anglerfish diversified into lots of different anglerfish, we kind of figured that the bacteria would be changing with them, right? So we expected for all of these different type of fish, we would expect to see all the different types of bacteria that live with them. If that's the case, then our next question would be, well, how do they get them? Because we don't know. Another thing people don't know about anglerfish is they have this really cool way of reproducing. They produce these big nets of this kind of mucosally tissue that they'll put out into the water that contains both egg and sperm. And then little baby anglerfish will stay in this lit part of the ocean until, just like a butterfly metamorphosizes into a the caterpillar metamorphosizes into a full-grown butterfly, that little tiny clear jelly bean of an anglerfish will metamorphosize into a dark anglerfish, but they usually will start swimming down first. They'll actually swim deeper than the adults are. And as they swim down, they'll metamorphosize and they'll get that darker color, and their lure will start popping out of their head. So again, we're wondering like how they get this bacteria, right? How do they get their lights lit up? Well, when does it happen? Does the mother anglerfish leave the bacteria? And I keep saying mother because only anglerfish that are female have lures. The males do not. So does the mother impart that bacteria onto that jelly net? And do we see bacteria that go all the way into the different life cycles? No, we don't. One of our collaborators, actually over at Nova University, found that the only place that we find these symbiotic bacteria are in the later stages in life. So it's after they've already undergone metamorphosis. So how do they get this bacteria? Especially this is complicated by the fact that these bacteria cannot live on their own. They have these genomes that have lost pieces to them. Often bacteria that are associated with organisms and do so very closely will start kind of, I'm anthropomorphizing, but they'll just like lose genes here and there because they don't need them anymore. It's kind of like maybe you forgot how to do laundry because your mom always did it for you, you know? So it's especially surprising that we don't see these anglerfish associated with the younger forms of life, and we only see them in later, because these bacteria can't just go out and swim on their own, or can they? What's interesting is that we expected to find lots of different bacteria, but we didn't. We only found 2 different types of bacteria associated with all of these different anglerfish. And we had multiple samples of different anglerfish. So we were able to establish basically that they are, and this is kind of like a sciencey thing that we do. If we match up how both of them are evolving, then they're evolving together. And if they aren't, then they aren't. So this immediately, and we see this all the time on land. Because we know that the bacteria actually are having to travel through the water, which is nuts, because really when you look at their genome, they can only eat sugar. So here's an example you can see here. So when we see host and bacteria, the picture, the trees on the right, these are just phylogenetic trees. When we see them line up all perfect like that, that means they're passed from parent to offspring. And when we see them kind of all tangled up, we actually call this a tanglegram. It means that they're being transmitted through the environment. And again, we've seen this a lot of times with parasites in humans and parasites in insects and other things. So this are the results. So for all these different anglerfish, we see really quickly that they're just a big, huge tangle. for both different types of species. So automatically we know that the bacteria are being transmitted through the environment. If you look at these bacteria under a microscope, you actually can see these like very anomalous little bodies that are actually hypothesized to be storage mechanisms. So what anglerfish are possibly doing to get from anglerfish to anglerfish is using these little storage molecules to be able to traverse the deep sea and try to find an anglerfish. And one of the really cool things, along with them being the most speciose deep sea fish, is that they're pretty spread out. And we only have 7 videos total of them in their natural environment. So these bacteria have to travel really long distances in really difficult conditions to get from anglerfish to anglerfish. But this is something we could have never figured out without using kind of these molecular tools and looking at the bacterial genomes. So it's really useful to be able to study this sort of stuff because the deep sea is expensive to study. And this is one way we can learn a whole lot about them using existing tools. Now, if I can just shift for a second, I use the exact same tools to study the bacteria that live on coral. I think most of the people who are on this call probably know that coral is not a rock. It's an animal, and this animal kind of looks like a little upside down anemone that makes its calcium carbonate shell. And living within that little animal are these symbiodinium, and the symbiodinium are what feeds the coral. This algae photosynthesizes and then gives that gives the food back to the coral, and the coral provides the algae a safe place to live. Well, in the spirit of a hat on a hat, along with the symbiodinium being a symbiont, the Rickettsia, these like bacteria, or RLO, or we also call them aqua Rickettsia, are living in the coral as well. But we think that they're actually not living in this like mutualistic interaction. They're probably interacting in a way that is detrimental, because they're not found to have anything that they give back to either the algae or to the coral, but they're actually taking stuff. They're not able to make their own sugars. They're not able to make their own ATP, which is one of those energy molecules. It's not able to make some of its own proteins. So what happens is that this bacteria will propagate and just love living inside the cell and eating all at once. But then when we see a bleaching event, like we're probably certainly going to see with these really hot temperatures outside, these bacteria leave the algae and it leaves the coral, and it becomes a much more sick system. And it basically leads that opens to the corals that harbor these bacteria are much more likely to get infected with things like white band disease. So I was brought on to do this study, and then the pandemic hit. And so what I had to do was go look for existing data sets. And luckily, a wonderful researcher had gathered all of these samples from all of the locations marked here on this map. And they had done it because they wanted to know about the genetics of coral and their symbiotic algae. But I was able to go through those existing samples and look for bacteria. What was awesome is we found right away, again, if you remember those tanglegrams I was showing you earlier, is that the bacteria are not differentiating similar to the coral animal, and they're not differentiating similar to the algae that they live with. They are more so just differentiating by location, which again is another indication that these bacteria are being transmitted horizontally, which just means not by the parent to the offspring, by some other source. What's also, because we can use, again, these genetic markers and these awesome new algorithms and genetic tools, we were able to determine that these aqua Rickettsia, these parasitic, these bad bacteria are replicating faster in Florida, and they're also evolving faster in Florida compared to the other locations where we were able to construct full genomes for them. This is likely due to the fact that the locations that they're at in Florida are ones that are more heavily impacted by human sources. So the bacteria is known using pictures of them under the microscope, we know that they're replicating, that they like to replicate faster in conditions where there's higher nutrients. So this makes studying these bacteria in Florida especially appealing, although it's also very frightening, of course, because we're going to get more bleaching, and these coral that are infected with this bacteria are more susceptible to disease. And disease is a big problem with coral in this area. So we're still kind of trying to work on the problem, as I said before. So they're not being transmitted by the algae, and they're not the algae that associates with the coral, and it's not being transferred by the coral animal. So we're looking to other hypotheses, and one of the big things that we're working on is possibly it's being passed from coral to coral through grazer interactions, so small animals that eat the coral. So not so much the fish, but we're thinking more of the smaller animals that can get caught in currents and stuff, or that it's being transmitted in the mucosal solution, because maybe the mucosal solution, that like weird gooey mucus stuff that like coral puts off, maybe that's nutrient rich enough to be able to transmit the bacteria because they can just live in that nutrient rich mucus. But we're still working on that. That's usually, you're doing science right when your question that you answer actually results in you having more questions. So you should be getting exponentially more ignorant as you go on. That's kind of our goal. Well, we try to figure out the questions that we have. But I always try to tell my students, and I think this is one of the things that underappreciated, because I think when people think about bacteria, they think it's kind of like dull and there's not a lot of potential there. But currently, I'm working on projects that are involved with understanding shark microbiome specifically. I have a collaborator here she goes out weekly and collects shark samples, and she does take blood. So she's now also taking gut samples for me, as well as studying some of the bacterial infections of lesions. And I get to take those samples and learn more about that. I want to continue on with working with bacteria associated with diatoms. I'm actually culturing some bacteria that we find on sharks that are bioluminescent and parasitic. So that's really fun. And then we get to use cool laser tools to figure out more about the bacteria that are growing on our coral. So there's a lot of potential. There's a lot of different things I get to work on, but that all kind of follow the same idea of it kind of being associated with these bacteria and understanding how bacteria, specifically symbionts, interact with animals. So that was kind of a quick overview, but I would love to hear any questions that anyone has about marine biology, bacteria, if you have any questions about what it's like to be a researcher at to be a lecturer at or to be one of my students, because I'm very proud of them as well. And at that last slide was highlighting some of the directions that they're going. So thank you for your time, and I look forward to answering questions.
Okay, thank you so much, Dr. Baker, for sharing your expertise on marine biology. And again, just like Dr. Baker said, we will take this time, so the audience can go ahead and enter their questions in the chat box. And while we're waiting for some of those questions to come in, I do want to share some additional library resources with our audience. And so the first one we have is Marine Biology, Cool Women Who Dive. It's by Karen Bush Gibson, which profiles 3 different women who work in marine biology. And the other one is The Lady and the Octopus by Dina Staff. It's about Jean Villepreux Power, who invented the first aquarium. Cool.
Wow, that's really cool.
Yeah.
It is one of those things. It's there was an ad that somebody shared on social media that was come be an oceanographer in the 1970s as a woman. You can take notes and clean glassware. So it shows you how far we've come since then.
Definitely. Oh, yeah.
So the question in the chat is, since there aren't a lot of anglerfish in the ocean, is there a lot of bioluminescent bacteria? just floating around. So we did look at samples of seawater and amplified a specific gene that is only found in our bioluminescent bacteria, and we did find that they were floating around. So we do know that in the case of anglerfish, those bacteria are just hanging around. But what the concentration is, it's really hard to be able to kind of slack back from that. But there's probably not that many because in theory, it only takes one bacteria to infect, to get into that lure and start setting up shop. And there aren't enough anglerfish around, so that undergoes what we call a bottleneck. Basically, an anglerfish will have its lure actually has an open hole to it and it will be leaking out those bacteria constantly. But only maybe 1 out of 10 to the 25th will get to another anglerfish. So there's lots of them out there, but they might perish before they find their host.
There was another question. Oh, did you see it? When they were asking how long is the marine biologist course?
So I mean, the course is just as long as a semester usually is. The NPS program that I run is an accelerated master's program. And the accelerated master's program only lasts a year and a half, which I think is a really great way for students, especially students who are already going to or going to school around the Miami area to get their master's degree, because we have a year of coursework, and the first part of it's kind of more, heavy duty, like math and ecology and all that stuff. And then the second semester is usually like fun, go play in the field, like get experience in whatever you're excited about. So yeah, it's, but all programs differ. So if you want to study it, if you want to get your undergraduate degree, there's lots of jobs in that. If you want to get your PhD, there's lots of jobs in that. So yeah.
And another question was, did you always want to study this?
Oddly, yes. I mean, I always tell students, especially like younger people, my mom was an English teacher in high school, and I was really good at English and history, and I was not as good in science. I really struggled to do well in the sciences, and I liked that struggle. I liked feeling like I had a lot to learn and that I wasn't just instantly good at something. And so I went to college for science. And that was a good decision, I think, because I think I bring a really unique perspective. But I wanted to start doing marine science, but the college I went to didn't have a marine science program. So I figured out I actually don't like dissecting things. So I started working on bacteria because it's like I don't have to dissect things. It kind of just happened organically. I know there's some people who like all they want to do is study like this thing. And I really like that studying bacteria, I can do like a lot of different stuff with it. Like you can see, like I have like an area of expertise that I can apply to lots of different stuff.
And the other question is, what college did you go to, and what was it like?
My undergraduate was at University of California, Berkeley. I graduated in 2005, and it was back in the day when I think we did a lot more like weeding out and kind of mistreatment of undergraduates because I felt very alone. I've since worked at Cornell as a lecturer part-time, and found that like a lot of schools are doing better at that. I had a really hard time as an undergrad. I was constantly worried that I wasn't good enough, and I was trying to work because I was trying to make enough money. But that being said, I did well enough I took time off, worked as a technician, and then I got my PhD at the University of Hawaii at Manoa, and that was excellent. There's nothing quite like being able to just go swimming and enjoy nature instead of stressing out. So I really liked that opportunity. There was a lot of independence. I got to study whatever I wanted to. And they pay you to get your PhD, which is one of the things my family, joked, like, they're like, oh, you could have been really great at this or that. And it's like, you all couldn't afford another year of college for me. So it's really nice that science PhDs are subsidized by research. So it was a much nicer experience. And so not only is your tuition covered, but you get a small stipend to be able to like do normal stuff like have a house and eat food and drive a car. But that's pretty much it. It's pretty austere. Anybody who thinks they're going into science for the money, there's not much there. But it's so rewarding. I've been to like 8 countries because of it, so like I can't complain.
Yeah, that experience alone makes up for it.
Yeah, it's really great.
And someone asked, isn't the string thing on top of the fish a light?
So the lure is lit by bacteria. But what's really weird, so some animals don't use bacteria, they can actually light themselves. And there are anglerfish where their lure on top of their head is lit by bacteria, and then they'll have these like long glowing, like almost looking like a beard. And that is not lit with bacteria. They light that themselves. So it's another question that we haven't been able to answer yet, why an animal would invest in having a bacteria do a function that in theory it could evolve to do itself. We don't know. There's always more questions.
And I think we have one more question, I believe, coming in.
It was just a thank you.
Thank you. I think that wraps up for the Q&A part. Again, Dr. Baker, thank you for joining us this evening and sharing your experiences with us. And thank you to our audience for all those great questions. If you do have any questions about our library programs or resources, just go to hcplc.org/contact. Where you'll find all our contact, including our phone number, which is 813-273-3652. And also, if you're interested in participating in our summer reading programs, online and in branches, you can get more information, again, at hcplc.org/events. Dr. Baker, do you want to go over, I know on here, it says that you can visit skypeascientist.com to learn more about this program, connecting scientists to classrooms, libraries, and families.
Mm-hmm. Yep, that's how we got connected. I've been doing Skype as Scientist since it originated. I think it was 4 years ago. So if anybody is an educator, or even if you do homeschool or have a homeschool group, or you again like libraries, penitentiaries, like any organization can request to have a science come in and chat. If you have any other questions for me, my Twitter is @Liddy, because I was an early adopter. You can also, if you Google Lydia Baker, University of Miami, you'll find my email address. Feel free to contact me if you have any questions about this or about our program or getting a master's degree, any of that stuff.
Thank you. Again, thank you, everyone, and I hope you all have a great night.
Thank you for the invitation.
Thank you.
Have a great one.